my NEET Cognition team doctors. Welcome back to day 4 of NEET Neural Lock series. Today we are going to do live for 30 minutes. This series is only for 30 minutes. Okay, it's only half an hour, but we are going to learn a lot. Today we are bridging the gap and going to tackle 3 massive blocks. That is block 1 endomembrane system, block 2 lysosomes, block 3 nucleus, nucleus and nucleoplasm. Yeah, okay. Remember the rule of this series, we don't just watch, we actively encode. That is, you have to open your notebooks, quickly get your pens, get ready to write every single high yield code word exactly 5 times with me to lock these NCERT marks straight into your memory. Okay, so a quick recap, okay, of what we saw for the past 3 days. In day 1 we did block scientist and discovery about cell theory, Schleiden and Schwann, omnicellular e-cellular, Rudolf Virchow. And in day 2 we did dimensions and size chart for the cells and prokaryote versus eukaryote structures. In this we saw what are all the organelles missing in prokaryotes, then about mesosomes. In block 2 we saw importance of prokaryotic examples. Can you name the prokaryotic examples? If you know the answer, quickly type it in the comment section. You have 4 examples in NCERT. Bacteria, blue-green algae, which is a sign of bacteria, then PPLO and mycoplasma. All those examples we saw with the importance. Next, yesterday what we saw is block 1 cell envelope. 3 layers of the cell envelope, that is glycocalyx, you have slime layer and capsule, cell wall in relation to staining, then plasma membrane, also called as selectively permeable membrane. Next is block 2, we saw ribosomes, 70S type in prokaryotes, made of 2 subunits, 50S plus 30S, larger and a smaller unit. S stands for Swedberg units, which is the sedimentation coefficient, an indirect measure of density and size. Next is the block 3, we saw about inclusion bodies. Can you name the examples of inclusion bodies? Phosphate granules, cyanoficin granules and glycogen granules. And today we are going to see 3 blocks, that is block 1 endomembrane system, block 2 lysosomes, block 3 nucleus, nucleus and nucleoplasm. So before going into the class, let me check the knowledge about all this. Okay, so first question, what are the organelles of endomembrane system? You can write the number of organelles, or you can, if you know the answer, that is the name of the organelles, quickly type it in the comment section. Second, lysosomes are formed from dash. From which organelle lysosomes are formed? And third, nucleoplasm consists of dash and dash. Okay, it consists of 2. If you know the answer, leave the answer in the comment section. Suppose if you don't know the answer, you wait. Once we complete the session, you type the answer and said you locked in. So now, shall we start? First is the block 1 endoplasmic reticulum, Golgi, lysosomes, vacuoles, forms endomembrane system. So what are the 4 organelles which forms the endomembrane system? Endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. Why these 4 organelles are involved? Because they have coordinated function. These are the code words which are used in NEET MCQs. Okay, coordinated function. Now, are you able to write it down without seeing? Endoplasmic reticulum, plus Golgi plus lysosomes and vacuoles. The fifth time, what are the organelles of the endomembrane system? Endoplasmic reticulum plus Golgi bodies plus lysosomes plus vacuoles. I think now you got the answer for the question which I asked in the opening, okay? Because all these organelles have coordinated function, that is each function is related to one another, but all the functions are different. That we will see in the video, okay? Now finally, what are the organelles which form the membrane system? Endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles. 4 organelles because they have coordinated function. Suppose if they ask you mitochondria, chloroplast, peroxisomes or any other choices other than this 4, you have to exclude it because they have independent function. Okay, we finished block 1. How many of you have completed? Okay, shall we move to block 2, lysosomes? Okay, they are also called as acid recyclers. Okay, they are called as acid recyclers. Now lysosomes are formed from what? Which organelle? Formed by packaging of Golgi apparatus. They are formed from Golgi apparatus, formed by the packaging of Golgi apparatus. Okay. Next is, what are all the enzymes seen in lysosomes? This is one very important MCQ. Hydrolytic enzymes. Lysis means, destruction, splitting, okay, and destruction. Next, active. All these enzymes are active under acidic pH. This is another MCQ. All these are MCQs. I've picked up only the words, okay, so that it will be easy for you to remember. So lysosomes are formed from Golgi apparatus by packaging. Lysosomes contain enzymes which are hydrolytic enzymes. They are destructive enzymes. All enzymes are active under which pH? Acidic pH. Get it now? So lysosomes are formed by packaging in Golgi apparatus. Lysosomes are formed by packaging in Golgi apparatus. Lysosomes are formed from packaging in Golgi apparatus. Lysosomes are formed from packaging in Golgi apparatus. All are hydrolytic enzymes. Can you name one of the hydrolytic enzyme of lysosome? If you know the answer, type the answer in the comment section quickly. Students, sometimes I may face a technical glitch. We may lose the connectivity. Meanwhile, you start memorizing all this. Okay, then I'll get back. Hydrolytic enzymes. Lysosomes contain hydrolytic enzymes. Next, all are active under acidic pH because they are what? Destructive enzymes. They are active under acidic pH. Lysosomes are formed in Golgi apparatus by packaging in Golgi apparatus. They contain hydrolytic enzymes which are active at acidic pH. They contain hydrolytic enzymes which are active at what pH? Acidic pH. You should never forget this, acidic pH. Very easy to remember. You remember the acids, okay, in the chemistry lab. It destroys, right? Lysis, destruction. So lysis means what? Destruction. Hydrolysis. So it's a destructive enzyme. Obviously it must be active in acidic pH. We finished block 2. Now moving to block 3. Students, we are We have concentrated till 14 minutes, and I'll ask you one question, okay? Have you seen most of the interviews about toppers and people who have many people have said, like even for the competitive exams and other exams, they say like how long they study. If you ask them, they study for 12 hours, 14 hours, 15 hours. Do you think is it possible? Is it possible? It's literally like, crushing your brain. It's really crushing your brain. So what you have to do? What you have to do? Okay, we'll see. Now I'm going to ask you one thing. Just a small break, okay? Focus versus time. Studying for 12 to 16 hours, you'll be mentally fatigued. You'll reach mental fatigue. Okay, you're using up all your fuel. There is no reserve. Whereas if you increase the focus, you try it once, increase the focus, just a deep work of 3 hours is enough. Okay, but at the if you're starting it for the first day, you can't do. Okay, for example, I'll say I was preparing for the live stream. Day 1, today is my 4th day. Day 1, even 2 days before, I started my preparation. But for the day 1, day 2, I took from morning. Day 3, I sorted out quickly. Today, day 4, it took me only 1 hour. Okay, so you cannot build focus at once. You have to get used to it. You have to get the practice. Okay, you have to do it daily. Make it as a habit. Focus should come as a habit. How you are able to focus a 2.5 hours movie? Try to remember. You'll remember each and every second of the movie, right? Which means you have very clear focus towards it, because it makes you feel interesting. Can you watch a movie for 16 hours? Are you watching it for 16 hours so that every scene gets locked into your mind? No. So focus is what you have to improve rather than increasing your time. Don't do 12 to 16 hours. I am a topper during my studies. Okay, I never did 12 to 16 hours. Then I won't go for the exam. Okay, it will totally spoil your health. So try to increase the focus. You know why you are able to finish, why you are not able to finish off your subjects, why you have backlogs? Because you frame a timetable without considering your what? Focus time. It's not time, it's the focus time. Okay, if you are able to think and make a logical timetable. Don't copy someone else. You can watch number of videos, number of timetables, but if you want to succeed, you have to work upon who yourself. See, I told you now, right? Maybe few people have seen most of my students, most of my classmates doing some 10 to 12 hours of study. Okay, but I used to do only 3 hours. That is morning 5 to 8. After a good night's sleep, I'll be feeling fresh, so I'll choose this 3 hours. Okay, it depends on the person. So I cannot see, what if I suppose blindly followed this 12 hours, because I scrutinized myself that how long can I maintain my focus. My focus is only for 3 hours, and that's enough for me. Okay, so work upon yourself, increase the focus time, not the time but the focus time. So from tomorrow, allot for deep work, that is only 1 hour. Okay, then gradually start increasing the time. Okay, so after the session is over, from tomorrow you are going to do this, and you are going to leave your ideas, okay, how you did that, in the comment section. Fine. So now we'll get back to block 3, that is the nucleus genetic command center. See, everyone is concentrating on the NEET marks, number of questions, but no one focuses on the mental health of the students. That is the primary thing which has to be fixed. Okay, they undergo a lot of stress because to bear the stress, they should be taught, they should be given proper instructions how to deal with the exam from the study timetable to daily hours, everything. Okay, we'll learn one by one. Now we'll get back to block 3, the nucleus, genetic command center. Then this is one important question. Okay, discovery of nucleus is by Robert Brown, 1831. I already told you in day 1, N means nucleus. Remember, okay, N to N. Robert Brown in 1831. I've seen students asking for some mnemonics, how to remember all this. The best thing is, sometimes if you keep too much of mnemonic, you will forget the mnemonic itself. That happened to me when I did my pharmacology second year. Okay, there will be a lot of medicines. If I try to keep the mnemonic, then I don't know for which I kept what mnemonic. Okay, so it's better sit down and write it as many times as possible, because this is life. Just to make it as a habit, I'm writing it for 5 times. But whenever you find any free time, you don't feel like studying, grab the notebook. Like now you have allotted a notebook for this, right? Make 5 columns and start writing. First is Robert Brown. So nucleus was discovered by Robert Brown in 1831. Who discovered nucleus? N for N. Robert Brown in 1831. Even you can circle and write so that you can remember this word automatically. Robert Brown, N for nucleus, 1831. This also we can find some clue, right? 1, both ends with 1. Then 83. If you have any idea, you can tell to me also. Robert Brown, I never used to do that. I always write it down. 1831. Robert Brown in 1831. Okay, next is nucleus. Where is nucleus? Where is nucleus? It is inside the nucleus. You have cell. Inside the cell, the command center. Okay, like how we have brain for our body, for a cell, nucleus is the brain. Okay, this is nucleus. Inside the cell, nucleus, inside the nucleus you have spherical structures. They don't have membrane. Okay, they are called nucleolus. Yes, singular. If it is one, you call it as nucleolus. If it is many, you call it as nucleoli. Nucleoli. Okay. This nucleolus, what is the function of nucleolus? Very important MCQ. Synthesizes ribosomal RNA, that is rRNA. You have how many types of RNA we have? 3 types of RNA. If you know the answer, quickly type it in the comment section. Okay. Ribosomal. Where do you study about mRNA, tRNA? Which chapter? At least you can mention the chapter. Okay, it comes in class 12. Ribosomal RNA synthesis. How do you feel this session, my students? Or do you feel the interaction? Because as I'm undergoing some withdrawal, feelings, recovery, I may leave touch with you. Okay, that's why I'm asking. Okay, we'll continue. Ribosomal RNA synthesis. You can leave your feedback in the comment section, whatever you feel. Okay, what is the goal of this team? NEET cognition. We have to make it as a good educational platform. Nucleolus, ribosomal RNA synthesis. What is the function of nucleolus? Ribosomal RNA synthesis. What is the function of nucleolus? Ribosomal RNA synthesis. And next, coming to this nucleoplasm. I asked you in the opening, What are the components of the nucleoplasm? Nucleolus plus chromatin. For that, first you should understand what is nucleoplasm. Nucleoplasm means you have cytoplasm, right? The fluid-like matrix. So inside the nucleus, you have some fluid-like portion. You call it as nucleoplasm. This nucleoplasm contains what? This nucleolus and the nucleoprotein fibers. They are called as the chromatin. So nucleoplasm contains nucleolus plus chromatin. If you don't understand, don't bother about it. You start writing the code words. During the class, we'll see it in detail. Okay? Because it's a short live, I'm focusing only on the code words. Nucleoplasm contains nucleolus and chromatin. Nucleoplasm contains nucleolus and chromatin. Nucleoplasm contains nucleolus and chromatin. Now, what is the other name of nucleoplasm? What is the other name of nucleoplasm? If you know the answer, type it in the comment section quickly. I'll tell you the answer. Okay, the nuclear matrix is also called as nucleoplasm. It is given in NCERT. Okay, nucleoplasm is also called as nuclear matrix. Now, shall we do a quick recap? So, this is block 1. In this, what are the organs of the endomembrane system? You have endoplasmic reticulum, Golgi apparatus, lysosomes and vacuoles, because all 4 have coordinated function. I will avoid these choices because they have independent function. They are no way related to endomembrane system. And next is the block 2, that is lysosomes. They are also called as acid recyclers. They are formed from Golgi apparatus by the process of packaging. Next, they contain hydrolytic enzymes, that is the destructive. Lysis means destruction, destructive enzymes, and they are active at acidic pH. Next, you have nucleus, the genetic command center. In this, the nucleus was discovered by Robert Brown in 1831. Then, nucleolus. The important function of nucleolus is ribosomal RNA synthesis. Then, nucleoplasm, which contains nucleolus and chromatin. What are the components of nucleoplasm? Okay, nucleolus and chromatin. Here we have one additional MCQ. Okay, a bonus MCQ, that is nuclear matrix is also called as nucleoplasm. I hope you enjoyed the session, students. Leave your feedback, and I hope you got the answer for the questions which I asked in the intro. If you know the answers, type the answers in the comment section, and all your feedback is very valuable for me. And thank you for listening and following me. Have a great day.